Hey everyone, I've got some really exciting news to share with you today. Researchers have made some major breakthroughs in the field of nanoscale endocytic processes, and let me tell you, the results are fascinating. These scientists used coarse-grained molecular dynamics simulations to study the behavior of nanovesicles when they come into contact with small liquid droplets. Can you believe it? They were able to uncover the morphological responses of nanovesicles and identify different pathways of endocytosis. And the best part? They found that the endocytic pathways are primarily controlled by two key factors: transbilayer stress asymmetry and line tension. These findings could have a major impact on the field of nanoscale endocytosis, so make sure you check out the full study. But wait, there's more. The researchers also showed that complete engulfment of the droplet is only possible for droplets that don't exceed a certain volume. So basically, they've uncovered some really important information about how nanovesicles and liquid droplets interact with each other at the nanoscale. Some questions and answers from this research work. What is the paper about? The paper discusses the morphological responses of nanovesicles when they come into contact with small liquid droplets and the endocytic pathways that result from this interaction. What are the key parameters affecting the endocytic pathways? The endocytic pathways are primarily dependent on two key parameters: the transbilayer stress asymmetry of the membrane and the line tension of the contact line between membrane and droplet. What can you learn from the paper? You can learn about the relationship between the lipid number null in the outer leaflet of a bilayer and the bilayer tensions, interfacial tension, and the pathways of endocytosis taken by a droplet. What is the general isoperimetric inequality from differential geometry used for in the paper? The general isoperimetric inequality from differential geometry is used to show that complete engulfment of a droplet by a nanovesicle is only possible for droplets that do not exceed a certain maximal droplet volume. If you're a science geek like me, you're definitely going to want to check out the full study. It's got all the details on how the researchers conducted their simulations, what they discovered, and what it means for the future of the field. So that's it for today, folks. Thanks for tuning in, and I hope you found this information as fascinating as I did. Don't forget to hit the like button and share this video with your science-loving friends. See you next time.